Every couple hopes to have a healthy child whenever they plan for a pregnancy. However, sometimes this does not happen and a child is born with a genetic disorder. There are more than 7,000 types of genetic diseases. The Department of Medical Genetics, established as a collaboration between Nizam's Institute of Medical Sciences and Center for DNA Fingerprinting and Diagnostics, has been functioning for the last 15 years, providing services in genetic diagnostics, genetic counseling, and therapy for some of these disorders. Recently, we worked collaboratively between the two institutes to work on a family where multiple babies were affected with an unknown genetic disorder, and our collaborative work resulted in a novel finding. I request my colleague, Dr. Shagun Agarwal, to tell you in detail about this finding.
A family attended the Department of Medical Genetics at NIMS Hospital in view of two of their babies having the same problem of water collection around the lung. So the first baby had died immediately after birth due to this, and in the second pregnancy, this problem was found at the 5th month. So we performed a detailed autopsy of this second baby to find out what was the problem, and we identified that this baby had multiple bubbles filled with water or fluid all over its internal organs. And we had never seen anything like this before, so we were strongly suspecting that both babies had some new previously unknown genetic disease. So we went ahead and studied the DNA of the second baby using next generation sequencing, and we were able to find that this baby's DNA had an error or a mutation in SERPINA11 gene. And this had previously never been described with any human disease. So we needed to further collaborate with the team at CDFT, and Dr. Rashna will be telling us about how we studied this further.
When Dr. Shagun approached us to help her understand whether this gene that she had uncovered, SERPINA11, was indeed the cause of the serpinopathy that she saw in the fetus, we really wanted to help. We first looked at the mutation that was there in the family and noticed that it would have led to a truncation of the protein so that a critical loop would not be present in this protein, the mutant protein. Next, we turned to mice, which are a very often used model system in basic research to understand the functions of human proteins. We noticed that mice expressed SERPINA11 in several tissues, including the lung, liver, heart, and kidney, all of which are tissues in which Dr. Shagun's team had found fluid-filled bubbles in the fetus. Then we noticed a very striking expression pattern for SERPINA11 in the mouse lung. It was expressed very highly in bronchioles, which are these narrow air passages in the lung. And this was quite unexpected because earlier reports had shown that SERPINA11 is only expressed in the liver. Finally, we were able to compare the lung sections of the affected fetus with a normal fetus. And in the normal fetus looks, the expression pattern looks very similar to that what we saw in mice. In the affected fetus, the SERPINA11 was clearly missing. What we want to do next is develop a mouse model by deleting SERPINA11 so that we can identify what are its targets and what are the molecular underpinnings of this novel lethal serpinopathy.
So with this, we were able to provide an answer to this family that the cause of their pregnancy losses was the SERPINA11 associated lethal disorder. And this can be prevented in the next pregnancies by going ahead with prenatal diagnosis.
The collaborative work between the two institutes resulted in identification of a novel gene disease association for the gene SERPINA11. This will help in development of diagnostics as well as therapy for conditions caused due to mutations in this gene in future. Our work was recently published in a reputed journal, Clinical Genetics, which is one of the respected journals in the field of medical genetics. In addition, the journal chose our work to be displayed on the cover page of the journal. We thank the directors of both institutes as well as the funding agencies like Department of Biotechnology and Department of Health Research, as well as Science and Engineering Research Board of the Department of Science and Technology for funding our research. We are very happy to have achieved this success and hope to achieve much more in future.